Yeah, when I first met with Nasha and some of the other parents from FOXG1, I was a little shocked because I would have felt like this disease was kind of well characterized for about 10 to 15 years, just their understanding of the biology, the understanding of the unmet medical need, the progression of disease, and really kind of the emerging level of epidemiology. I felt like the organization had such a level of professionalism similar to those that have been around for many, many years, that I was extremely impressed and excited to work with them. What I would also say is that clearly that level of impression went far and beyond just me, because every time I would either talk to an investor or a collaborator or a friend in the industry, they all would say, "Hey, I heard you guys are working on FOXG1, or have you heard of FOXG1? It's similar." Kind of similar to REC, it could be an interesting target to go after. Yeah, you guys should talk to FOXG1 Foundation. That happened like 3 times. And so ultimately it was one of those things that said, hey, we better get on top of it, because these guys are moving forward. So I was actually extremely excited and impressed when I first met with leadership. Yeah, I think for me, it starts with the understanding of the biology of the disease, and it starts with having an animal model where you could actually test the efficacy and the safety of a potential therapeutic. If you don't have that, you're kind of lost, lost in this land of ambiguous discovery. And the fact of the matter that these guys have done such amazing work in really such a short period of time. The disease and the understanding of the disease is just starting to kind of really be elucidated. And the fact of the matter that the availability of an animal model, and the understanding of the biology of the gene in question is just far and exceeds a lot of diseases that have actually been discovered but never really fully characterized. And ultimately, as a company, that's what we need to really understand how to be able to best target it. Understanding, is it just a simple gene replacement? Is this a knockdown approach? Do we have to have somewhat of a nuanced approach where we have to guard against overexpression, which it looks like that's kind of what we'll need to do with FOXG1. And just kind of, we have the tools in the toolbox, but it's really all about putting those tools in the right order together in order to appropriately address the underlying cause of the disease. And the only way you do that is having an understanding of not only the biology, but the burden of illness and how the disease manifests itself in the real world. And you only get that from the patient perspective. Most importantly, you get it from the parents who are living it every day, and to have that drive to go and be able to find a cure. And so, again, for me, I think having the disease well characterized as it is, having the availability of an animal model, I think is just really important steps that usually take patient advocacy groups decades to get to, and this has been done in such a short period of time. I'm really impressed by that progress. The word cure is a tough word to use, right? And it means something different for everybody, to be quite honest. In our early days when we were working on SMA, where you have such a terrible disease where it's primarily 100% mortality by 20 months of age, and to be able to see a child that's treated early enough, essentially identified in utero, treated 3 weeks, and to basically, even at this stage in life, now, that child is probably 4 years old or so. Just the fact that he's lived beyond his second birthday is a miracle, but really have thrived and essentially living a life of a normal child shows that the technology is there. But the biology of every disease is different. I think what's going to be most important, and this is really where the patient advocacy groups can help us, is around early diagnosis. Early treatment and early diagnosis is always going to be is always going to be better. I think when you start to think about diseases like FOXG1, which are neurodevelopmental, there is probably some level of reversibility. And there's probably a high level of reversibility in these diseases because the patients aren't losing neurons, or neurons aren't dying off like in our neurodegenerative diseases that we're going after, where time is really neurons. In the case here, there's just something that's not allowing the neurons to be fully functional, or is something that is cutting down on the connectivity of one neuron talking to the next, right? And so if we can restore that level of connectivity, restore the functional neurons that are there because they're there to be restored, I do think there's a high degree of reversibility, similar to kind of what I think is starting to be well elucidated from, similar diseases like Rett syndrome, where that is starting to really be understood. What I would say is I think the combination of early treatment and a level of reversibility, then you can start using the C word, as we call it, because we don't even really say that internally. That's our goal, though, right? When I went into gene therapy and to be able to see the curative nature of gene therapy, this is the reason why I don't work in small molecules. It's the reason why I don't work in conventional biologics, because you can't see a miracle and then go back to doing what you were doing before. And so ultimately, that's what we're trying to do. We're trying to take this technology that's worked in similar diseases and apply it at scale to diseases that look like those, right? And so people ask us, will we go after liver diseases or systemic diseases or cardiac diseases? And my short answer is, well, Taysha may do that, but RA won't be here. But because I feel strongly that there's enough diseases for us to be able to go after with the technology that we know extremely well, that's worked in our own hands, that could apply it to a number of these other cases. And, hopefully FOXG1 will be a good example of that. Yeah, I think, primarily what I would say, what ultimately got me interested in the early days, I think now the biology of the disease on its own, and honestly, just the patient community, they're like the coolest patient community. I mean, just really cool people, right? So, and what I would probably say early on, it was probably the similarities to Rett syndrome that got us interested, because you have to, our academic founders, Dr. Stephen Gray, and the people that worked in his lab, including Sarah Sennett, who are now on their own doing just amazing things. It took them 15 years to work out Rett. And it's not because they weren't trying, but this whole odyssey of understanding the underlying biology of the disease. Just replacing the gene wasn't enough. If you replace the gene, you then found out that, Rett syndrome, when overexpressed, is toxic, right? So then that created another issue. And so could you do it through dosing, trying to titrate dose? Well, you can't, because these genes essentially transduce whatever cells that they hit. And having this notion of mosaicism and Rett syndrome made it extremely difficult. Essentially trying to make the healthy cells healthy and not make the sick cells sick, right? That's a very narrow window to strive for, and we've essentially been able to do it. But it was 15 years in the making, and Taysha just came around at the right time. FOXG1 has some similarities. In some ways, it's a little bit easier, right? In the case where, you don't have this whole level of Goldilocks, but you do have a level of overexpression, right? Overexpression can be bad. That's something that we can deal with, right? That's something that we can deal with, because that's a similarity to diseases like Angelman. That's similarities to diseases like Fragile X, where a little you may be able to overshoot a little bit, but there is a point where you get to too much, and that can cause some sort of toxicity. And in some cases, it's a more attractive target than a lot of the diseases that we're going after. And I think, fortunate for us, and in our approach to FOXG1, some of the tools that we've developed to essentially guard against overexpression associated toxicity in a genotypic manner can be used for this particular disease. And so that's essentially what we're trying to do. And because the diseases are very similar in around the way that the gene functions, we feel like the same target panel, our MI-rare target panel, should work. At least that's that's our initial thought. And so that saves us amount of time in order to be able to go directly into animal models versus trying to go find new, down-regulatory microRNAs to then build into the transgene. So for us, it makes it a really attractive target. And like I said, the FOXG1 community is just a cool patient community to work with. It's such a great question, and hopefully, I think when you look 15 years down the road, we've achieved at least partially achieved our mission from some of these diseases, which is eradicating monogenic CNS disease. And I use the word eradicate. It's a rough word. It's kind of a nasty word, to be kind of honest. And we had a lot of debate when we were coming up with this mission, and that word eradicate, because it's harsh. And you know, we had a number of people said, "Oh, you shouldn't use this word." Some of the marketers say you shouldn't use this word, and I basically said these diseases are nasty and harsh. So why not use a harsh and nasty word? And I think as soon as I said that, people kind of got it. And so for me, I want to make some of these diseases look like polio, right? Where it's just gone. Where, you know, 10 years from now, 15 years from now, 20 years from now, there's a kid that said, you know, when I was born, my mom told me I had something called FOXG1. I really don't know what that was, but you know, they told me I need to kind of just stay on top of it. But yeah, you know, yeah, you know, I don't know what it means. That's the goal, right? And to be able to do that for diseases like FOXG1, Rett, you know, Angelman, GM2, CLN1, CLN7, right? Just to apply this technology that we know works extremely well, but to really do it at scale for a number of diseases. But that's something that I take away. That's my goal. Our chief medical officer, Suyash Prasad, who's a pediatrician and he's a pediatric intensivist. So he's a pediatric ER doctor, right? And he's seen the worst of the worst. That's what he always says. He said, I just want to see a kid that natural history told you should look totally different. And they just walk up to you in the restaurant one day and you have a conversation and they find out you're in genetic medicines and they say, yeah, I think I had a genetic disease or, you know, but yeah, but we don't really talk about it. Yeah. You know, that's cool. That's what we're going for.